In this video, we will be going through proteins, which is a part of the biological molecules topic. If you haven't seen the carbohydrates and lipids videos, head over to my channel and give them a watch. So proteins are called polypeptides as well, which are the polymers. Amino acids are the monomers, which make up proteins. There are many examples of proteins within cells, like enzymes, channel proteins in membranes, hormones, or immunoglobulins. For your exam, it is important that you are able to draw out the general structure of amino acids as well as know how to label the different groups on them. As you can see in this picture, you have the carboxylic acid group, the amino group, which has the nitrogen atom, and the variable R group. It is this R group which changes each amino acid. There are 20 amino acids, so 20 R groups. The bond that forms between two amino acids is called the peptide bond. This covalent bond forms by removing a water molecule, the hydroxyl group from the carboxylic acid group, and the hydrogen from the amino group. This is done by the condensation reaction. In order to reverse this and break the peptide bond, this is done by adding a water molecule via a hydrolysis reaction. In your exam, it is important that you are able to join amino acids together and highlight the peptide bond. However complicated the R group may look, take it back to the basic, generalized structure of an amino acid and add the R group in last. To test for proteins, you need to use the Biuret reagent. If proteins are present, the solution will change from blue to lilac. However, this only detects if there are more than one peptide bond, so it does not detect amino acids and dipeptides. You also need to know about the structure and function of proteins. The amino acid sequence is what determines the types of bonds that can form, which will determine the protein shape and hence will determine the protein's function. There are 4 levels to protein structure that you need to know about. First up is the primary structure. This is the foundation of the protein structure. It is the amino acid sequence bonded together by peptide bonds. The secondary structure can either be an alpha helix or beta pleated sheet, which is where the primary structure has been folded with hydrogen bonds to form these structures. The hydrogen bonds can form between the weakly negative charged oxygen atom or nitrogen atom to the weakly positively charged hydrogen atom. The tertiary structure is what forms the 3D shape of the protein. The secondary structure is further folded with extra bonds depending on the R groups, like more hydrogen bonds, ionic bonds, disulfide bridges, which form between the amino acid cysteine and weak hydrophobic interactions. Finally, we have the quaternary structure, which is only present in proteins with more than one polypeptide chain. The 2 examples that you need to be aware of for comparison questions in your exams are haemoglobin and collagen. Haemoglobin is a type of globular protein. It is a spherical protein with 4 polypeptide chains. It has 2 alpha subunits and 2 beta subunits. Now, be careful. Something to be aware of is in your exam if they have fill in the blank questions and ask you to pick from the options below. When you are in a rush, do not get confused with alpha helix and beta pleated sheets, which are secondary structures, and alpha and beta subunits, which are parts of haemoglobin in the quaternary structure. Each polypeptide chain has a prosthetic heme group, which contains an iron to ion. Haemoglobin is soluble as the hydrophilic R groups are facing outwards and the hydrophobic R groups are facing inwards. Haemoglobin's function is to transport oxygen around the blood within the red blood cells and become oxyhemoglobin. 1 oxygen molecule can bind to each heme group on each polypeptide, so 1 haemoglobin can carry 4 oxygen molecules. Collagen is a type of fibrous protein. It is formed from 3 polypeptide chains which twist together to form a helix and held in place by hydrogen bonds. Every third amino acid in collagen is made from glycine, so the amino acid sequence is quite repetitive. Collagen is insoluble. It is a structural protein used in connective tissue found in tendons and ligaments. Again, for your exam, it is important that you link how the structure enables the protein's function for both haemoglobin and collagen. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.